We live in a microbial world, and a microbial world lives on and within us. Our bodies are constantly being exposed to bacteria, fungi, parasites, and viruses and must restrict the normal flora from entering into sterile tissue sites, discriminate between friend and foe, and defend against invading microbes. Our body's defenses are similar to a military defense. The initial defense mechanisms are barriers such as the skin, acid and bile of the gastrointestinal tract, and mucus that inactivate and prevent entry of the foreign agents. If these barriers are compromised or the agent gains entry in another way, the local militia of innate responses must quickly rally to the challenge and prevent expansion of the invasion. Initially, toxic molecules, defensins and other peptids, complement, are thrown at the microbe while other molecules make them sticky, complement, lectins, and antibodies, facilitating the ingestion and destruction of the microbe by neutrophils and macrophages. Once activated, these responses also send an alarm, complement, cytokines, and chemokines, to other cells and open the vasculature, complement, cytokines, to provide access to the site. If these steps are not effective, the innate responses activate a major campaign specifically directed against the invader by antigen-specific immune responses, B cells, antibody, and T cells, at whatever cost, immunopathogenesis. Finally, the infected tissue must be repaired and the system returned to the status quo and a normal regulated balance. Knowledge of the characteristics of the enemy, antigens, through prior exposure or vaccination enables the body to mount a faster, more effective response, activation of memory B and T cells, on re-challenge. The different elements of the immune system interact and communicate using soluble molecules and by direct cell-to-cell interaction. These interactions provide the mechanisms for activation and control of the protective responses. Unfortunately, the protective responses to some infectious agents are insufficient or too slow. In other cases, the response to the challenge is excessive and causes peripheral damage. In either case, disease occurs. Soluble activators and stimulators of innate and immune functions. Innate and immune cells communicate by cell-cell interactions and with soluble molecules, including complement cleavage products, cytokines, interferons, and chemokines. Cytokines are hormone-like proteins that act on cells to activate and regulate the innate and immune response. Interferons are also cytokines that are produced in response to viral and other infections, interferon IFN, alpha and IFN, or on activation of the immune response IFN. They promote antiviral and anti-tumor responses and stimulate immune responses. Chemokines are small proteins, almost equals 8,000 Da, that attract specific cells to sites of inflammation and other immunologically important sites. Neutrophils, basophils, natural killer, NK, cells, monocytes, and T cells express receptors and can be activated by specific chemokines. The chemokines and other proteins, for example, the C3a and C5a products of the complement cascade, are chemotactic factors that establish a chemical path to attract phagocytic and inflammatory cells to the site of infection. The triggers that stimulate the production of these molecules, and the consequences of the interactions with their receptors on specific cells determine the nature of the innate and immune response. Useful mnemonics to help learn cytokines are stat, source, cell, trigger, action, target, receptor and cell, and for the response, TikTok, trigger, inducer, cells, producer and responder, time course, outcome, cytokines. Cells of the immune response. Immune responses are mediated by specific cells with defined functions. For each of the cells know the CARP, cell surface markers, for example, CD4, TCR, etc., actions, kill, suppress, activate, etc., role, type of response, products, cytokines, antibody, etc. The white blood cells can be distinguished on the basis of 1. Morphology, 2. Histologic staining, 3. Immunologic functions, and, for intracellular and cell surface markers. B and T lymphocytes can be distinguished by expression of antigen receptors on their surfaces, immunoglobulin for B cells and T cell receptors for T cells. Other cell surface proteins distinguish subsets of these and other types of cells. These marker proteins are identified with monoclonal antibodies. They are defined within clusters of differentiation, as determined by all of the monoclonal antibodies that recognize the same molecule, for example, CD4, or group of molecules, for example, CD3, and the markers indicated by CD, cluster of differentiation, numbers. In addition, all nucleated cells express class I MHC, MHCI, antigens, human, HLA-A, HLA-B, HLA-C. A special class of cells that are antigen-presenting cells, APCs, express class II major histocompatible complex, MHC, antigens, HLA-DR, HLA-DP, HLA-DQ. Cells that present antigenic peptides to T cells include dendritic cells, macrophage family cells, B lymphocytes, and a limited number of other cell types. Hematopoietic cell differentiation. Differentiation of a common progenitor cell, termed the pluripotent stem cell, gives rise to all blood cells. Differentiation of these cells begins during development of the fetus and continues throughout life. The pluripotent stem cell differentiates into stem cells, sometimes referred to as colony-forming units, for different lineages of blood cells, including the lymphoid, T and B cells, myeloid, erythrocytic, and megakaryoblastic, source of platelets, lineages. The stem cells reside primarily in the bone marrow but can also be isolated from the fetal blood in umbilical cords, and as rare cells in adult blood. Differentiation of stem cells into the functional blood cells is triggered by specific cell surface interactions with the stromal cells of the marrow and specific cytokines produced by these and other cells. The bone marrow and thymus are considered primary lymphoid organs. These sites of initial lymphocyte differentiation are essential to the development of the immune system. The thymus is essential at birth for T cell development but shrinks with aging, and other tissues adopt its function later in life. Secondary lymphoid organs include the lymph nodes, spleen, skin, and mucosa-associated lymphoid tissue, MALT. The latter also includes gut-associated lymphoid tissue, GALT, for example, Peyer's patches, and bronchus-associated lymphoid tissue, BALT, for example, lung. These sites are where dendritic cells and B and T lymphocytes reside and respond to antigenic challenges. The cells of the primary and secondary lymphoid organs produce chemokines and express cell surface adhesion molecules, addressins, that interact with homing receptors, cell adhesion molecules, expressed on B and T cells. The spleen and lymph nodes are encapsulated organs in which the macrophages and B and T cells reside in defined regions. Their location facilitates interactions that promote immune responses to antigen. Proliferation of the lymphocytes in response to infectious challenge causes these tissues to swell, that is, swollen glands. The lymph nodes are kidney-shaped organs, 2 to 10 mm in diameter, that filter the fluid that passes from intercellular spaces into the lymphatic system, almost like a sewage processing plant. The lymph node is constructed to optimize the meeting of the innate, dendritic cells and macrophages, and the immune response, B and T, cells to initiate and expand specific immune responses. A lymph node consists of 3 layers. 1. The cortex, the outer layer that contains mainly B cells, follicular dendritic cells, and macrophages arranged in structures called follicles and, if activated, in germinal centers. 2. The paracortex, which contains T cells and dendritic cells, which present antigens to the T cells to initiate immune responses. 3. The medulla, which contains B and T cells and antibody-producing plasma cells, as well as channels for the lymph fluid. The spleen is a large organ that acts like a lymph node and also filters antigens, encapsulated bacteria, and viruses from blood and removes aged blood cells and platelets. The spleen consists of 2 types of tissue, the white pulp and the red pulp. The white pulp consists of arterioles surrounded by lymphoid cells, periarterial a lymphoid sheath, in which the T cells surround the central arteriole. B cells are organized into primary unstimulated or secondary stimulated follicles that have a germinal center. The germinal center contains memory cells, macrophages, and follicular dendritic cells. The red pulp is a storage site for blood cells and the site of turnover of aged platelets and erythrocytes. A hint for remembering this is, there is no T in follicle or germinal center, but there are TS in paracortex and periarterial lymphoid sheet. The epidermis of skin contains Langerhans cells, and the dermis contains dendritic cells, B and T lymphocytes, macrophages, and mast cells. Large numbers of memory T cells continuously circulate into these layers of the skin. Keratinocytes in the epidermis are part of the innate antimicrobial defense system. MALT contains less structured aggregates of lymphoid cells. For example, the Peyer's patches along the intestinal wall have special cells in the epithelium, M cells, that deliver antigens from the lumen into a mini lymph node, like structure containing dendritic cells and lymphocytes in defined regions, T, interfollicular, and B, germinal. Dendritic cells, T cells, and B cells also reside in the lamina propria layer just under the epithelium. Once thought to be expendable, the tonsils are an important part of the MALT. These lymphoepithelial organs sample the microbes in the oral and nasal area. The tonsils contain a large number of mature and memory B cells, 50% to 90% of the lymphocytes, that use their antibodies to sense specific pathogens and, with dendritic cells and T cells, can initiate immune responses. Swelling of the tonsils may be caused by infection or a response to infection. MALT contains less structured aggregates of lymphoid cells. For example, the Peyer's patches along the intestinal wall have special cells in the epithelium, M cells, that deliver antigens from the lumen into a mini lymph node, like structure containing dendritic cells and lymphocytes in defined regions, T, interfollicular, and B, germinal. Dendritic cells, T cells, and B cells also reside in the lamina propria layer just under the epithelium. Once thought to be expendable, the tonsils are an important part of the MALT. These lymphoepithelial organs sample the microbes in the oral and nasal area. The tonsils contain a large number of mature and memory B cells, 50% to 90% of the lymphocytes, that use their antibodies to sense specific pathogens and, with dendritic cells and T cells, can initiate immune responses. Swelling of the tonsils may be caused by infection or a response to infection. Polymorphonuclear leukocytes. Polymorphonuclear leukocytes, neutrophils, are short-lived cells that constitute 50% to 70% of circulating white blood cells, and are a primary phagocytic defense against bacterial infection and major component of the inflammatory response. Neutrophils are 9 to 14 m in diameter, lack mitochondria, have a granulated cytoplasm in which granules stain with both acidic and basic stains, and have a multilobed nucleus. Neutrophils leave the blood and concentrate at the site of infection in response to chemotactic factors. During infection, the neutrophils in the blood increase in number and include precursor forms. These precursors are termed band forms, in contrast to the terminally differentiated and segmented neutrophils. The finding of such an increase and change in neutrophils by a blood count is sometimes termed a left shift with an increase in bands versus segs. Neutrophils ingest bacteria by phagocytosis and expose the bacteria to antibacterial substances and enzymes contained in primary, azurophilic, and secondary, specific, granules. Azurophilic granules are reservoirs for enzymes such as myeloperoxidase, glucuronidase, elastase, and cathepsin G. Specific granules serve as reservoirs for lysozyme and lactoferrin. Dead neutrophils release a sticky antimicrobial net of DNA and other fibers, termed neutrophil extracellular trap, NET, and dead neutrophils are the major component of pus. Eosinophils are heavily granulated cells, 11 to 15 m in diameter, with a bilobed nucleus that stains with the acid dye eosin Y. They are also phagocytic, motile, and granulated. The granules contain acid phosphatase, peroxidase, and eosinophilic basic proteins. Eosinophils play a role in the defense against parasitic infections. The eosinophilic basic proteins are toxic to many parasites. Mast cells and basophils are non-phagocytic granulocytes that release the contents of their granules in response to inflammatory triggers, and during allergic responses, type 1 hypersensitivity. Mononuclear phagocyte system. The mononuclear phagocyte system has myeloid cells and consists of monocytes, C, in the blood, macrophages, and dendritic cells. Monocytes are 10 to 18 m in diameter, with a single-lobed, kidney bean-shaped nucleus. They represent 3% to 8% of peripheral blood leukocytes. Monocytes follow neutrophils into tissue as an early cellular component of inflammation. Macrophages are long-lived cells that are phagocytic, contain lysosomes, and unlike neutrophils, have mitochondria. Macrophages have the following basic functions: 1. Phagocytosis of debris and microbes. 2. Antigen presentation to T cells to expand specific immune responses, and 3. Secretion of cytokines to either maintain normal tissue function and repair, M2 macrophages, or be antimicrobial and promote inflammation, M1 macrophages. Macrophages express cell surface receptors for the Fc portion of immunoglobulin Ig, Ge, FcRI, FcRII, FcRII, and for the C3b product of the complement cascade, CR1, CR3. These receptors facilitate the phagocytosis of antigen, bacteria, or viruses coated with these proteins. Toll-like and other pattern recognition receptors recognize pathogen-associated molecular patterns and activate protective responses. Macrophages also express the class II MHC antigen, which allows these cells to present antigen to CD4 helper T cells to expand the immune response. Macrophages secrete interleukin, Illinois-1, Illinois-6, tumor necrosis factor, TNF, alpha, Illinois-12, and other molecules upon sensing bacteria, which stimulates immune and inflammatory responses, including fever. A T cell, a derived cytokine, IFN, activates inflammatory functions in macrophages. Activated macrophages, M1, have enhanced phagocytic, killing, and antigen-presenting capabilities. Blood, alveolar, and serosal, for example, peritoneal, macrophages are examples of wandering macrophages derived from bone marrow precursor cells. Tissue-resident macrophages include alveolar macrophages in the lungs, Kupffer cells in the liver, intraglomerular mesangial cells in the kidney, histiocytes in connective tissue, osteoclasts, synovial cells, and microglial cells in the brain. These macrophages are descended from cells of the yolk sac and are primarily involved in tissue maintenance and repair functions. M2 macrophages. The mature forms of these cells have different morphologies corresponding to their ultimate tissue location and function, and may express a subset of macrophage activities or cell surface markers. Dendritic cells. Most dendritic cells have octopus-like tendrils and are professional antigen-presenting cells, APCs, that can also produce cytokines. Different types of immature and mature dendritic cells are found in tissue and blood. They include Langerhans cells in the skin, dermal interstitial cells, splenic marginal dendritic cells, and dendritic cells in the liver, thymus, germinal centers of the lymph nodes, and blood. Plasmacytoid dendritic cells have a plasma cell-like appearance, are present in blood, and produce large amounts of IFN and cytokines in response to viral and other infections. Dendritic cells may be derived from stem cells or monocytes. Immature dendritic cells capture and phagocytose antigen efficiently, and release cytokines to activate and steer the subsequent immune response. Upon maturation, dendritic cells move to lymph node regions rich in T cells to present antigen on class I and class II MHC antigens. Dendritic cells are the only antigen-presenting cells that can initiate an immune response with a naive T lymphocyte, and they also determine the type of response, Th1, Th2, Th17, Treg. Follicular dendritic cells localized to B-cell regions of lymph nodes and spleen are not hematopoietic in origin, and do not process antigen but have tendrils, dendrites, and a sticky surface to concentrate and display antigens to B cells. Lymphocytes. The lymphocytes are 6 to 10 m in diameter, which is smaller than leukocytes. There are three classes of lymphocytes: T cells, B cells, and innate lymphoid cells. These cells have a large nucleus and smaller, a granular cytoplasm. Although B and T cells are indistinguishable by their morphologic features, they can be distinguished on the basis of function and surface markers. Lymphoid cells that are not B or T cells are termed innate lymphoid cells, ILCs, and include the NK cells. T cells acquired their name because they develop in the thymus. T cells have the following two major functions in response to foreign antigen. 1. Regulate, suppress, when necessary, and activate immune and inflammatory responses by cell-to-cell interactions and by releasing cytokines. 2. Directly kill virally infected cells, foreign cells, for example, tissue grafts, and tumors. By promoting apoptosis, T cells make up 60% to 80% of peripheral blood lymphocytes. T cells were initially distinguished from B cells on the basis of their ability to bind and surround themselves, forming rosettes, with sheep erythrocytes through the CD2 molecule. All T cells express an antigen-binding T cell receptor, TCR, which resembles but differs from antibody, and CD2, and CD3 associated proteins on their cell surface. T cells are divided into three major groups on the basis of the type of TCR and also by the cell surface expression of two proteins, CD4 and CD8. Most lymphocytes express the alpha-beta TCR. CD4 expressing T cells are primarily cytokine-producing cells that help initiate, direct, and regulate innate and immune responses. The CD4 T cells can be further divided into Th0, Th1, Th2, Th17, Treg, and other subgroups according to the spectrum of cytokines they secrete and the type of immune response they promote. Th1 cells promote local antibody and cellular inflammatory responses, whereas Th2 cells promote antibody production. Th17 cells activate epithelial cell and neutrophil-driven inflammation and other responses, and Treg cells regulate the immune response to maintain balance and tolerance of self. The CD8 T cells also release cytokines but are better known for their ability to recognize and kill, through apoptosis, virally infected cells, foreign tissue transplants, non-self grafts, and tumor cells as cytotoxic T cells. T cells also produce memory cells that express CD45RO. A variable number of T cells express the TCR but do not express CD4 or CD8. These cells generally reside in skin and mucosa and are important for innate immunity. NKT cells are T cells that share characteristics with NK cells. The primary function of B cells is to make antibody, but they also internalize antigen, process the antigen, and present the antigen to T cells to request T cell help and expand the immune response. B cells can be identified by the presence of immunoglobulins, class II MHC molecules, and receptors for the C3b and C3d products of the complement cascade, CR1, CR2, on their cell surfaces. The B cell name is derived from its site of differentiation, the bursa of Fabricius in birds and the bone marrow of mammals. Activated B cells either apoptose, develop into memory cells, which express the CD45RO cell surface marker and circulate until activated by specific antigen, or terminally differentiate into plasma cells. Plasma cells have small nuclei and a large cytoplasm for their job as producers of antibody. Innate lymphoid cells, ILCs, are non-B, non-T lymphocytes that resemble T cells in some characteristics and include the NK cells. Cytokine-producing ILCs are found associated with epithelial cells in the thymus and in the intestines. In the gut, these cells produce cytokines that regulate the epithelial cell and T cell response to the intestinal flora, and facilitate antiparasitic worm protection. Errors in their function are associated with immunopathology, including autoimmune diseases. ILCs are also involved in regulating immune responses during pregnancy. The large, Granular lymphocyte NK cells resemble the CD8 T cells in cytolytic function toward virally infected and tumor cells, but they differ in the mechanism for identifying the target cell. NK cells are also capable of antibody-dependent killing and hence are also called antibody-dependent cellular cytotoxicity, ADCC or K, cells. The cytoplasmic granules contain cytolytic proteins to mediate the killing.